Dear student, today I will teach you Unit 3, which is Cell Structure and Functions. So the Unit 3 contains Chapter 8, which is Cell, the unit of life. For this, I am following NCERT Class 11th Biology book. So what is cell? Cell is the fundamental structural and functional unit of all living organism. So how it is fundamental structural? So we know human bodies are made up of organs, and these organs are made up of tissues, okay, and tissues are made up of cells. So cell is the basic unit of life, basic structural unit of life, and these cell contain several components which perform different kind of function which is necessary for the function of life. So one cell contain all the component which is required for function of life. So that is why cell is called fundamental structural and functional unit of all living organism. So there are two type of organism. One is unicellular organism and other are multicellular organism. Unicellular organism have example like paramecium, amoeba, bacteria, yeast because they are made up of single cell and contain all functional unit which is necessary for their normal function. Okay, and multicellular organism, we know there is example of insect, human body and plant body because they contain so many cells which perform different kind of function, and these function are necessary for their life. Okay, so cells are the fundamental structural and functional unit of living organisms, and they are of two type of organism. One is unicellular and other is multicellular organism. So if we consider unicellular organism, you can see there is only single cell, so they can have independent existence, and they contain all necessary component which perform function of life. So unicellular organisms are capable of independent existence and they can perform all the essential function of life. Okay, so Anton von Leeuwenhoek, he was the first person who saw and described the life cell, and Robert Brown, he later discovered that there is nucleus present inside the cell. So what is cell theory? In 1838, Matthias Schildin, he was a German botanist. He examined large number of plants and observed that these plants are composed of different kinds of cell, and which form the tissue of plant. So in 1838, Matthias Schildin, he studied different kind of plants and found that they are made up of different kind of cell which further form tissue of the plant. And Theodor Schwann, he was the zoologist. So he studied different kind of animal cell and reported that these cell, they contain a outer layer which is known as plasma membrane. And later he said that plant tissue contain cell wall. So here you can see animal cell, they have only plasma membrane, and plant cell they have plasma membrane plus cell wall. Okay, so here you can see in this figure there is animal cell, plant cell. In animal cell there is only cell membrane. Here you can see there is only cell membrane, but this cell membrane is also present in plant cell, but cell wall is present only in plant cell and it is not present in animal cell. So this is the difference between animal cell and plant cell. So Sheldon and Schwann together formulated the cell theory, but this theory has a drawback because it was not giving any explanation that how new cell were formed. So Rudolf Virchow in 1855 he explained that cell divide and new cells are formed from pre-existing cell, means there are some pre-existing cell which further divide to give a new cell. So he modified the theory of Schildin and so on and composed new theory, which is first point is all living organisms are composed of cells and product of cell. So there are living organism, they have a structural basic structure unit cell, and further these cells combine together they form tissue. So all living organisms are composed of cells and product of cell. And second point is all cells arise from pre-existing cell. So Rudolf Virchow just included this part in the existing theory of Schildin and Schwann, which were saying that all organisms are composed of cell and product of cell. Here in this figure you can see this is pre-existing cell, and now it is getting divided and giving you two cell. So all cells are made up of the pre-existing cell. So in class 7th and 8th, maybe you have seen onion cell and chick cell under the camera. So in onion cell you may have seen there is cell membrane and cell wall, and each cell there is one circular component which is called nucleus. And similarly you have seen in chick cell there is plasma membrane, and in each cell there is some circular component, dotted component, that is called nucleus. Okay, so inside each cell there is a dense membrane bound structure called nucleus, and these nucleus contain the chromosome, which in turn contain the genetic material and DNA. So inside each cell there is a nucleus, and this nucleus contain genetic material. So they are present in the form of chromosome. So here in this figure you can see this is nucleus, and inside nucleus you can see green thread like component, like you are watching this one. This is chromosome. And if you study smaller part of this chromosome, you can call it is DNA. And if you further study some smaller segment, that is called gene. So cells are of different size and shape. So first we will consider shape. So here you can see red blood cell. Red blood cell are circular and they are biconcave. And white blood cell are amoeba shape. And here you can see cell in the column form with nucleus. They are long and narrow. They are epithelial cell. Here you can see branch and long chain type cell. So these are nerve cell. Here you can see this is kidney shaped cell, which is also called guard cells. So these are of different shapes. So now we consider size factor. Mycoplasma, the smallest cells, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. And the largest isolated single cell is the egg of ostrich. And among multicellular organisms, human red blood cells are 7 micrometer in diameter, and nerve cells are some of the longest cells present in human body. So cells are of different shapes and size. Now we'll study overview of cell. Cells that have membrane-bound nuclei, they are called eukaryotic cell, whereas cells that lack a membrane-bound nucleus are called prokaryotic cell. For example, this is a cell. Inside this cell there is a nucleus, and this nucleus, if they contain a membrane outside, then these cells are called as eukaryotic. Okay, eukaryotic. If there is another cell which contain nucleus but that nucleus is not membrane bound, then these cells are called prokaryotic cell. Okay, and another thing, in both prokaryotic and eukaryotic there is a semi-fluid matrix is called cytoplasm. So inside this cell there is some fluid present. I am showing this fluid in dotted form. So this fluid is called cytoplasm. Okay. So this cytoplasm is the main arena of cellular activity. So whatever cellular activity happens, it happens in this semi-fluids portion. Okay. And so in eukaryotic cell, other than cytoplasm, there are other membrane-bound organelle, which is endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. In prokaryotic cell, these organelles are not present, but in prokaryotic and eukaryotic, there is ribosome present. Okay, so in prokaryotic cell, only ribosomes are present, but in eukaryotic cell, there is ribosome plus these organelles are present. So within cell, ribosomes are found not only in cytoplasm but also within two other organelle, which is chloroplast and mitochondria. So this is not only present in cytoplasm, this semi-fluid portion is cytoplasm. So these ribosomes are not only present in cytoplasm but they also found in chloroplast and mitochondria, which are two organelles. Okay, and animal cell contain another non-membrane bound organelle which is called centrosome. So this help in cell division. Okay, so here in this overview of cell we study what is a eukaryotic cell and what is prokaryotic cell and what is cytoplasm and eukaryotic cell contain what type of organelles. Organelles you can take it as a component of cell. Okay, and what kind of organelles are present in prokaryotic cell. So in this class we will study only prokaryotic cell. In next class we will study eukaryotic cell. Prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma and PPLO. They are generally smaller and multiply more rapidly than eukaryotic cell. So these are prokaryotic cell. You can see bacteria has size 1 to 2 micrometer and PPLO is a size of 0.1 micrometer. So virus are in the size of 0.02 to 0.2 micrometer. When you compare this prokaryotic cell with eukaryotic cell has the size of 10 to 20 micrometer. Okay, so these prokaryotic cell can be of different size and shapes. Now if we consider shape factor, in bacteria they are of bacillus rod type. So you can see this is rod type. So these rod type cells are called bacillus, and when it is in spherical shape they are called coccus. So here you can see this is spherical shape, they are called coccus, and coma shaped bacteria are called vibrio. You have heard the term Vibrio cholera, Staphylococcus, Bacillus, and also Spirillum means spiral shape. So if bacteria is of spiral shape, they are called spirillous. Okay. So the organization of prokaryotic cell is fundamentally similar even though prokaryotes exhibit wide varieties of shapes and functions. So doesn't matter they have different size and shape, their fundamental function are very similar. Now we will study more about prokaryotic cell. In prokaryotic cell there is a cell wall which surrounds cell membrane except in mycoplasma. So this is figure of bacterial cell. In bacterial cell you can see this blue color outer layer which is called capsule. Inside this capsule there is one layer this whole layer, this is called cell wall, and this cell wall surround inner layer which is called plasma membrane. So this cell wall is not present in mycoplasma and remaining it is present in all kind of prokaryotic cell. Okay, now the inside this cell there is fluid matrix which is called cytoplasm. Okay, so this fluid matrix area, you can see these dotted. These are some kind of fluids present. This is called cytoplasm. Okay, and there is no well-defined nucleus. Nucleus is not well defined because it is not present inside any kind of membrane. So the genetic material is naked. In addition to genomic DNA, many bacteria have small circular DNA outside the genomic DNA. These small DNA are called plasmid. Okay. So other than genomic DNA, there is some circular DNA present which is called plasmid. Okay. So you can call there is some plasmid present inside the cell which is circular DNA. Okay. So this is circular DNA. This is circular DNA which is called plasmid. Okay, the plasmid DNA confers certain unique phenotype character to such bacteria. So it provides phenotypic character to bacteria. One such character is resistance to antibiotic. So this plasmid help bacteria from certain medicine which kills bacteria. So they help in providing resistance toward and various antibiotics. So nuclear membrane is found in eukaryote. I already told you prokaryotes contain nucleus without nuclear membrane, but eukaryotes have nucleus and this nucleus has nuclear membrane outside. In a prokaryotic cell, there is no organelle present, but only ribosomes are present. And same ribosomes are also present in eukaryotic cell. But eukaryotic cell contains so many organelle I told you like endoplasmic reticulum, Golgi apparatus. Okay, but in prokaryote these kind of organelles are not present, but only ribosomes are present. Prokaryotes have something unique in the form of inclusion. So here you can say further extension of plasma membrane here. This further extension is called mesosome. Okay, so this is unique feature of prokaryotic cell. So these mesosome they are essentially infolding of cell membrane. So now we will study more about cell envelope and its most modification. So most prokaryotic cell, particularly the bacterial cell, have a chemically complex cell envelope. Okay, so they have complex cell envelope. The cell envelope consists of tightly bound three-layered structure. So these three-layer structure is capsule, cell wall, and plasma membrane, which I showed you in previous slide. Okay, so capsule this capsule is made up of glycocalyx, okay, which made the outer portion. Inside this glycocalyx there is cell wall, and inside this cell wall there is a plasma membrane. So these three layers they have different function, but they act together as a single protective unit. So on the basis of this cell wall, bacteria are further divided in two categories. One is gram positive and other is gram negative. So gram is the name of scientist who created one dye, one staining reagent. When any bacteria attain this reagent and become colored, they are called gram positive, and bacteria who don't get stained they are called gram negative bacteria. So now we'll come to glycocalyx portion. If this glycocalyx portion is loose sheet, they are called slime layer, and if they are very tight, compact, they are called capsule. So these glycocalyx calyx are of two type. One is slime layer when it is loose sheet, and when it is very tight, they are called capsule. And the cell wall determine the shape of cell and provide strong structural support to prevent the bacteria from bursting or collapsing. Okay, and the plasma membrane is permeable and it interact with the outside world. A special membranous structure is the mesosome, which is formed by extension of plasma membrane. I already told you this is extension of plasma membrane, and this is called mesosome. The extension is in the form of vesicle. Vesicle are like circular shape. If it is circular shape, then it is called vesicle. Sometime it is in tubule form and sometime it is in lamellae form. So they help in cell wall formation, DNA replication, and distribution to daughter cell. So when one cell divide to another cell, other cell is called daughter cell. So this mesosome, they perform the function of cell wall formation, DNA replication, and distribution to daughter cell. They also help in respiration, secretion process to increase the surface area of plasma membrane and enzymatic content. So in this paragraph, I told you about the function of this mesosome. So in some prokaryotes like cyanobacteria, there are other membrane extension in the cytoplasm called chromatophore, which contain pigment. So chrome means pigment. So chromatophores which contain pigment. So other than mesosome, there is one more extension which is called chromatophore in cyanobacteria. This is only present in cyanobacteria. So bacterial cell may be motile or non-motile, means they can move or they cannot move. If they are motile, they have thin filamentous extension. So you can see this filamentous extension. So you can consider it as a tail of bacteria. When this tail waves, this bacteria moves. So these filaments help in motility of bacteria. So this whole unit is called flagella. This flagella has three components. One is filament, I told you this tail portion. Other is hook portion. Hook means here they are joined with the cell, and other is basal body. Okay. The filament is the longest portion which extend from the cell surface to the outside. So now here we are studying about tail of bacteria. Tail help in motility, and this tail has a three component, and this tail is called flagella as a complete body. So this flagella has a three component. One is filament, which is this long portion. Other is hook. This is the hook by which it is attached to the cell, and a basal body. Beside flagella, there are pili and fimbriae also present. This is pili. They are smaller than this flagellum, but they are bigger than fimbriae. Fimbriae is also present in some bacteria like this, like this, like this, like this. So pili is also helps bacteria in movement, and they are tubular structure made up of special protein. Okay, and if fimbriae are present, they are small bristle-like fibers, and this help bacteria to attach to rock in stream and also to host tissue. So fimbriae has role in attaching bacteria to rocks or some host cell. Now this is the last slide. regarding prokaryotic cell, and I already told you prokaryotic cell contain one organelle which is called ribosome. Apart from ribosome, we will study inclusion bodies. So in prokaryotic cell, ribosomes are associated with plasma membrane of cell. They are 15 nanometer and 20 nanometer in size and made up of two units, which is 50S and 30S. When these units are present together, they form 70S unit. So you may be confused why not 50 and 30 is making 80S. It is not like in biology. When you go in higher class, you will study why it is 70S unit. So just at this point, learn 50S and 30S unit. When they are present together, they form 70S ribosomes. Okay. So what is the function of ribosome? Ribosome help in synthesis of protein. And when these ribosomes, when they get attached to single mRNA, they form a chain of polyribosome or polysome. Then these ribosome or polysomes translate the mRNA into protein. Okay. So here you can see there is 30S subunit of ribosome. This is 50S subunit of ribosome. Means this blue portion is 30S and this green portion is 50S subunit of ribosome. So when these two ribosome they get attached to this mRNA chain, you can see this long red chain which is 5' to 3' and this is mRNA chain. When so many ribosomes attached on this mRNA chain, they form polyribosome or polysome. So what is their function? They translate this mRNA into amino acid chain. So their main function is amino acid synthesis from mRNA chain. Okay. Inclusion body. So whatever reserve material in prokaryotic cells present, they are stored in the cytoplasm in the form of inclusion bodies. They are not bound by any membrane system and they lie freely in cytoplasm. For example, phosphate granules, cyanophosphine granules, and glycogen granules. So gas vacuoles are found in blue-green algae and purple and green photosynthetic bacteria. So here I will finish part 1 of cell, and today we study what is cell, what is the eukaryotic cell, what is prokaryotic cell, and we studied in detail about prokaryotic cell. In next class we will study what is eukaryotic cell. Thank you for attending the class.